So this one is topic 4. We're going to talk about the nucleic acids. So nucleic acids and proteins. There is DNA and RNA. These are what do the information that is encoded, heritable information, structural functions. So proteins are really involved in what DNA, RNA sequences are like because they are directly related to protein synthesis based on DNA, RNA. Different structure, have different functions. So let's just move on to nucleic acid. Nucleic acids are made out of 3 things, which is pentose, phosphate group, and nitrogenous base. And there's 5 sugar for pentose, either be ribose or deoxyribose. If it's if it's missing an oxygen, then it's going to be a deoxyribose. Phosphates have charge, they're negatively charged phosphate group on the number 5 carbon. And the nitrogenous base are the ATC or ATU or C or G, and we're going to be talking about those as well. So nucleotides are sugars, like I said, and the only difference really is the OH and the H. They are 1, 2, 3, 4, 5. So they're pentose. There are 5 carbons. And over here, nitrogenous base, pyrimidine are cytosine, thymine, and uracil. And purines are adenine and guanine. So remember it as pure Ga, pure gold, not gold exactly, but Ga, and cut the pie, you know, CUT, so cut the pie, PYR, and purines are pure gold, GA, random, really random. So PUR, purines and pyrimidines bind, pyrimidine and purines bind, so these guys bind together, either one of them will bind, so it's either uracil or thymine actually, because the uracil is only for RNA and thymine is only for DNA. They're anti-parallel, meaning opposite direction. So one of them is going one way and the other one is going the other way. I'm not really exactly sure what that direction is supposed to do. There's hydrogen bonds between these guys, and there is only 2 hydrogen bonds between the A and the T. So between, AT has 2 bonds and CG has 3 bonds. Just remember that. Oh yeah, also the fact that they're like double ring or single ringed. So single ringed for pyrimidine and double ringed for purines. Pyrimidines are small, CUT are small, purines are big. So deoxyribose are called the deoxyribonuclease. There's a gene that is specific. Yeah, it's over here. Nucleotides, DNA, RNA. There's genes. A discrepancy in gene can cause a lot of damage. It can cause diseases and stuff. And there's chemicals written in the nucleic acid code that must be decoded into proteins. So gene expression, if coded in the gene, transmit into gene. So whatever that's in the gene will be transformed into the product, because RNA is going to be turned into a protein, right? Separation of DNA to program them. Structure, these structures are written chemical code. Okay. Deoxyribonucleate double-stranded. Not yet. Okay, we're gonna come back to these, I think. Functions transmit info into info to build proteins. Genes determine the protein sequence, etc. Okay, so now we're at phosphodiester bonds. So phosphodiester bonds are really strong. They're very, very strong. They're what make the sugar phosphate backbone, and they're resilient to heat and to pH. And this is what makes DNA itself so stable. Confer polarity direction to DNA strands, negative phosphate group at 5 and sugar at the 3 end. So at the 5 end, there's a phosphate group, and sugar at the 3' end group. covalent bonds stronger than hydrogen bonds. Yeah, so phosphodiester bonds are stronger than hydrogen bonds, which are the bonds that are making up the purines and pyrimidines. There's dehydration synthesis to break these phosphodiester bonds, and those are called nuclease. Nuclease is a type of enzyme that can break this, which is dehydration synthesis. Yeah, those are called the sugar phosphate backbone of this polymer. So phosphodiester bonds can be dehydrated. Yeah, so now they're double helix, so you can talk about it here, I think. Double helix, they're anti-parallel, meaning one is going one way, the other one's going the other way. Two strands held by hydrogen bond between these, like I said, anti-parallel double helix. And we can calculate the percentage of the Ag and Tc, right? Because if there is, for every A, there's a T. And if they give you the percentage of A, well, there's the same percentage of T. So then 100 take away the percentage of A and T. And G and C will be the remaining. So cut that into half and we can find it. However, if it is a uracil, well, then that does not make any sense because uracils are only on RNA. They do not double strand. Gene is a function segment of DNA that provides genetic information necessary to provide. So gene, the necessary information to make protein. DNA stranded with protein is called chromatin. So when DNA strand is in association with protein, they become chromatin. And chromatin gets folded into chromosomes, where human has 46. Human have 46 chromosomes. DNA strand associated with protein is called the chromatin. Gene expressions when a gene is transformed into product, which is a protein. So why is it double-stranded? Double-stranded for more information, less space. Gene located in the either strands. So either strands, there's gene location. Stable structure because it's double helix. Very stable structure. One fewer OH on ribose hydroxyl group on the ribose sugar. So that's the DNA quality. So they have one fewer OH, which makes them somehow more stable. And protect blueprint from the damage. So this helix shape will make them more stronger and more stable and resilient to damage. So now talking about RNA, consists of subunits called ribonucleotides. So the DNA is made out of nucleotides, and the RNAs are ribonucleotides. I forgot about that word. And they have resonance of thymine. They are now adenine to uracil. They're making multiple kinds of RNA with different functions. So messenger RNA for messages. rRNA are catalytic activity for ribosomal RNA, which is inside the nucleolus. And there is a transfer tRNA. Single-stranded can be folded into 3D. They will leave the nucleus and they will synthesize from the DNA. They're synthesized from the DNA. And they have the OH on their ribose. rRNA are very versatile. They can do a lot of activities. So it's used as blueprints for some viruses. RNA could be used for some blueprints for some organisms like viruses. RNA, DNA is synthesized from DNA nucleus. Yeah, so RNA is synthesized from DNA and it will leave the nucleus to be synthesized into protein and stuff. Now there's this little table which explains about the CG and AT pairing. They're in the nucleus. They're double-stranded, double helix, long-term. and they're more stable. RNAs C, G, and AU, nucleolus, and cytoplasmic, single-stranded, and they form 3D molecule. They're short-term and they're less stable. They're more reactive. Now let's move on to proteins. Proteins, all process of life for 50% organic matter in the body. Their main organic matter in the body, typical animal body, and play important role in all of matter of life, and 50% of organic matter is made out of proteins. Yeah, it's also about their shape, and their shape will change their functions. So there's something called the amino acid, which is right here. The R group could be proteinated, meaning can become looking like a very huge molecule here. So this just R just to simplify it, I think. And amino acids are the monomers of proteins. They will make polypeptides. Yeah, amino acids is made by each amino acids will be bonding to form protein, but amino acid chain, I should say. Polypeptides. Yeah, it's polypeptides. So yeah, linear chain of those are called polypeptides. I didn't write it. Oh, I did write it. Polypeptides. So these are bonded by peptide bonds, and that's between the carboxyl group and the amino group. And there's the alpha group, monomers of units of proteins, and shape, order, and sequence will change their function, and protein shape will influence the function. There's hydrophilic and hydrophobic amino acids. So looking at the hydrophilic ones, they are a lot CH like this. There's a lot of CH like this. There's one that is a ring, but that one's special. Yeah, most of them that are hydrophilic, these amino acids, there's 20 of them, is looking like these chains of carbon, whereas the hydrophobic ones seem to have a lot more, well, not a lot, but they're a bit more branched, and they're a bit more carbon ringed. Between the cytosine and the final shape of the hydrophilic nonpolar shape of protein determines functional chemical property. So essential amino acids, where is that? Where's my essential amino acids? Well, essential amino acids are basically just ones that human cannot produce. That's why need to be consumed from food. That's what it means for anything that says essential. So there's dipeptide and polypeptide. Dipeptide is basically 2 of the aminos, and polypeptide is a chain of amino acids. There's the condensation reaction of H2O to produce these polymers. Peptide bond between amino and carboxyl group, like I said. Condensation reaction, yep, that's fine. Amino acids, proteins. Yeah, so each group will form one water molecule. So by counting the water molecule, we can count how many bonds were made, and from that we can deduce how many amino acids are making this chain, polypeptide chain. Polypeptide chain. This is called the polypeptide chain. Polypeptide chain. 20 to 35,000 amino acids is the length of proteins. It depends and can vary from 20 to 35,000. And they have many structures. There is the primary structure, secondary structure, tertiary structure, and these different structure level will build up our proteins. The first one is determined by the DNA or inherited information, and that's the unique sequence of amino acids. So each individual amino acid will make these chains like this. And they can change from hydrophilic to hydrophobic. If one of them change, can just change all of the rest of the structures, the next levels. Secondary is coils, alpha helix coils, or folded beta pleated sheet. It's the second level of the structure. And then there is another layer of structure called the tertiary structure, which is what determines the 3D, which gives us 3D shaped, 3D shapes determined by R groups. H bond, ionic bond, van der Waals, disulfide bridge, very strong covalent bonds. There's the pH and the heat that there are that can denature, or I should say, can break these. Then we can then make them come apart and no longer function. So those are some factors that can denature proteins. And structures are due to R group, one polypeptides. Quaternary is the last level, and some can stop at the tertiary, but the quaternary is when 2 or more polypeptide chains form a macromolecule. So a polypeptide chain, like I said, is the amino acid chains. 2 or more forming gives a quaternary structure of molecules, macromolecules. Good example is hemoglobin, where there is 4 polypeptides that make up this globular protein. 4 subunit tertiary structure will make up hemoglobin, which is one of our blood cell proteins. So one single change in this nucleotide will just make everything go bad and can cause diseases. So now going on to structure of the proteins, there's the structure for function of the proteins, structure support like keratin and collagen for skin and tissue, storage, amino acids, store amino acids, and chysin, breakdown of amino acids. So like in eggs, there's lots of chysin. Transport for transport proteins that can make them go across the membrane, let them go in, transport. There's a lot about transport later on. Hemoglobin, oxygen transport, signaling, and hormones respond to stimuli, detect stimuli, hormone coordination, receptor movement, so muscle protein, motor proteins, defensive antibodies against disease, virus, bacteria, and enzymatic selective acceleration, chemical reaction, hydrolysis of the bond, and these are all catalysts. Yeah, I went over like 10 slides in like one breath right now. Okay. So, the denaturation happens to the tertiary structure, but also secondary, but mainly tertiary. They will actually break them down all the way to unfold them all the way to the chain structure, the primary structure. They will lose the functional activity and cause disease such as Alzheimer's, done by denaturation of these proteins. And the denaturation can be caused by pH or temperature or the chemical substance around them. Some folding significant diseases are cystic fibrosis, Alzheimer, Parkinson's, Creutzfeldt disease, Creutzfeldt-Jakob disease. These are all diseases from the denaturation. And there's also renaturation, which is going back to it, which is rare. It doesn't happen all the time. But most proteins can be unfolded or denatured by these factors. And that's it for the 4th chapter.